I also work on the executive committee of computational biology and bioinformatics, where I talk to and collaborate with Mark Gerstein. And this module of the course for CBB 752 will be focused on physical modeling of biological systems and simulation of biological systems. And the examples I'm going to draw from are proteins. So there are many different types of proteins: membrane proteins, intrinsically disordered proteins, globular proteins. For the first part of the course, I'm going to talk about the simpler types of proteins, globular proteins, and I'm going to talk about their structure. There was, so here's the schedule. It's a little bit revised. So today, which is April 19th, and Wednesday, April 21st, we're going to talk about protein structure, focusing on globular proteins. And then there'll be a special lecture because I missed the last lecture. So it'll be on Thursday, April 22nd at 1 o'clock. And then we'll also on Monday, April 26th, we'll talk about intrinsically disordered proteins, which have connections to different disease states and aggregation. And then the final lecture will be on April 28th, a Monday, and we'll talk about molecular dynamics simulations, how to simulate protein structure and dynamics. And if you have any questions, you can go ahead and just interrupt at any time if you want to ask a question, or if you're listening to this at some other time, you can send me an email. Happy to answer questions. So just a first sort of cartoon of what proteins are. So proteins are examples of biological polymers. So a polymer is a string of monomers that are linked together via chemical bonds. And the interesting thing about polymers is they can have many different types of structures. So in the top image, I show a linear polymer, which is in an extended state. And then I show on the right, there's a protein in a folded state. And so this proteins, which are polymers, can transition between very different types of states, one that's extended or one that's more compact and that has a type of order. It sort of goes without saying that proteins are important in biological systems. So just two examples, they're very important for catalyzing biochemical reactions, transporting molecules across cell membranes, and there are numerous other examples. In the case of proteins, I said it was a polymer. So a peptide is sort of a special noun that means a small protein. So that would include tens of amino acids, whereas a protein has hundreds or sometimes even thousands of amino acids. And so the monomers for a protein are the amino acids. So you're going to string these together to form a longer chain. There are 20 different types of amino acids. And so we'll talk about those. And since there are 20, there are so many different types of amino acids, you can get lots of different sequences. And the sequences would be putting those 20 amino acids in different orders. So a very simple structure would be all of the amino acids were the same, and so they were all, say, alanine. And then that would be a polyalanine protein. But you can obviously create 20 to the n different amino acid sequences if you have n amino acids together. So as I said before, one very interesting thing and why biology makes use of polymers is they can have very different states and structures. So the cartoon of proteins is that when they're first created, they're in this unstructured, unfolded state, and then to perform particular biological functions, they need to be in a very specific, particular structure, which is typically much more compact and ordered than the unfolded, unstructured state. Most current experimental and computational studies of proteins have been in very idealized environments where the solvent is very simple and the protein concentrations are very dilute, which is not necessarily physiologically relevant for how proteins occur in cells. And so a very interesting, important upcoming field is how protein structure changes when they're in the cellular environment. So in the test tube, what you can do is you can change the structure of proteins, and a lot of experimental studies do this and computational studies do this. You can unfold a protein by increasing the temperature or adding chemical denaturants, and this is a way to understand the folding transition in proteins. So proteins have a typical stability, which is at what temperature does it fold or unfold, and the same thing with its solvent quality. So a first slide on some of the sort of chemical aspects of the structure. So this gives you some insight into what amino acids are. So amino acids are made up of an N-terminal group, so a nitrogen, and a C-terminal group, a carbon, which is in a carboxyl group. And then there's a central, what's called an alpha carbon in the center, off which a side chain branches off. And in this particular diagram, it's called R. And so amino acids share the amine group and the carboxyl group and the alpha carbon, but then different amino acids have these different variable groups which branch off of the alpha carbon. And so each of the 20 amino acids differ by that side chain. And so the amino acids bond together through peptide bonds. And so on the right, I show an example of a glycine, alanine, and a leucine, where they're bound together with a peptide bond between the carboxyl carbon and the nitrogen of the amine group. And then it begins the discussion of the side chains, and you see glycine there has a very simple side chain, just a hydrogen coming off the central C alpha carbon, whereas alanine has the CH3 group for its side chain, and then leucine has a even more complicated side chain coming off. And those side chains are very important for determining the structure of proteins. So here's a slide with the complete set of amino acids, and they're broken up into nonpolar, polar, and electrically charged. So the nonpolar are residue amino. So also, I think I've already done it now. Oftentimes I will interchangeably switch amino acid and residue. So they mean the exact same thing. So an amino acid and residue in my mind can be switched. So don't let me confuse you. And so I have nonpolar, which don't like to be adjacent to water molecules, polar groups, which can like to be near water molecules, and many of these residues in the polar group can hydrogen bond. And then electrically charged residues which can be either slightly positively or negatively charged. The electrically charged and the polar amino acids tend to be found on the surface of the protein, as we'll see later on in these slides, whereas the nonpolar amino acids tend to be buried in the central region of the protein, which I call the core. And we'll see that as well by measuring what's called the solvent accessible surface area of a protein and or the amino acids. And so here you can see what I mean by what differentiates different amino acids. So you see they all share what's called the backbone, which is this amine group, the alpha carbon, and the carboxyl group. And then they differ by the side chain. As I said before, you see glycine there is very simple, alanine, and then as you move to the right, they increase in their complexity. So definitely in the 70s and 80s, and to this day, there's something called the protein folding problem. And what it is that it's this idea of, sequences, you can come up with amino acid sequences, and you can ask, what's the structure associated with that amino acid sequence? And the idea is that proteins don't have sort of arbitrary amino acid sequences. They most likely take on sequences that through evolutionary processes are foldable into particular structures that are associated with function. And so there's this thought that sequences are not random, but if I do have a particular sequence, what is the structure associated with that unique sequence? So the protein folding problem is what unique folded 3D structure of a protein is determined by that particular sequence. Right about that time, when people were thinking about this sequence to structure problem, people realized that if a system has many degrees of freedom, and we're talking about hundreds or thousands of degrees of freedom, and what I mean by a degree of freedom is that in the simplest case, it would be, well, where do all the atoms, where should they all be situated? How does the protein know where to put all of the atoms that are associated with the amino acids? Or in the case of proteins, we'll see that the degrees of freedom that are relevant are the angles. So the relative angles between 3 different atoms and the angles between 4 sequential atoms. And so those are the degrees of freedom, and the protein has to decide, well, what are those angles? What do I set for those degrees of freedom so I can uniquely specify the structure? And so the idea is, well, if there are so many possible ways to sort of construct or to set those degrees of freedom, then how can the protein find the exact set of values that those angles should be? And so there's a little sort of back of the envelope calculation that was originally due to Leventhal. And so I'll just go through that now. So I'm going to imagine that there's a protein with n amino acids. And then you can talk about for that n amino acids, you have to specify what the conformations of those amino acids are. And the main conformations for those amino acids are what are called dihedral angles. And we're going to talk in great detail about what dihedral angles are. But just for now, just say that those amino acids are going to be in some conformation, and you have to specify that conformation. Are they going to be sort of extended? Are they going to be somewhat compact? And so So there are two dihedral angles for each amino acid. So that's what the 2 is in that formula. And mu represents the number of possibilities for that dihedral angle. So it turns out that the dihedral angles can't take on a continuous number of values. They typically only take on 3. So usually, negative 60, 60, or 180. So they're called rotamers, and we're going to see that in great detail as well, that dihedral angles don't typically take on a continuum of values. They take on typically a few different values. And so the number of conformations for n amino acids where each dihedral angle can take on 3 particular values, and there are 2 dihedral angles per n amino acids. It turns out that that's 3 to the 200. So there are 3 to the 200 possible conformations, a very conservative guess, for what the possible number of conformations of a protein chain. And so 3 to the 200 is very large. It's an astronomical number. It's approximately 10 to the 95. And what you can do is you can say, well, how does a protein know what conformation to pick? And so what you can say is what it does is it goes into a conformation, resides in that conformation for a little while, and then it decides, well, this is not the right conformation for me. I'm going to leave. And so it's this idea that it's going to explore each of those conformations. And so if you just say that the exploration time per one of those conformations is a picosecond, just a very small time scale, and you plug in 10 to the 95 times a picosecond, you still get an astronomical number. So the folding times for at least fast folders, protein folders, are a microsecond to a millisecond, whereas Leventhal's paradox gives this astronomical folding time if the protein had to sample all of the these fake or bad conformations that it shouldn't be in. And so that's Leventhal's paradox. It sort of assumes a random sampling of an energy landscape. So the idea for any complex system, whether it's biological or physical, is the idea of an energy landscape. And so what it says is that every conformation of the system, so for example, every set of locations of the atoms corresponds to a value of the energy. And so then that means that the energy of the system can be viewed as a surface in a very high dimensional space. So in 2 dimensions, it turns out it's this sequence of minima, maxima, and saddle points. And so what I'm just drawing here is the horizontal axis represents the positions of the atoms, or equivalently the angles, conformations of the dihedral angles. And so it's just saying that there's a line. So at every point along this line corresponds to a configuration of the atoms, and there's an energy associated with every possible position of the atoms. And this is just a two-dimensional rendering, it's really a 3n dimensional rendering if you have n amino acids. And so the idea is that there are minima, maxima, and saddle points, and any physical or biological system wants to exist in the lowest energy structure. And so the protein folding problem is trying to navigate your way through this energy landscape to find the the lowest energy minimum and not getting stuck in a minimum that's higher than the lowest energy minimum. So in this cartoon, M1 represents the lowest energy minimum, M2 and M3 are minima, but they're slightly higher. And so in various model systems, especially in computation, to some extent in experiments, what people have done is tried to map out the energy landscape for protein systems. And what they think is that proteins, at least those that have gone through evolution, significant evolutionary pressure and round after round after round of selection, is that the energy landscape for proteins, or well-folded proteins, looks like the one on the left and not the one on the right. So that the one on the left is what's called a funneled landscape, where the roughness is very weak. So that, and roughness is sort of what it means is that the sort of root mean square fluctuations in the energy are fairly small, and you can't get trapped, and it's very difficult to get trapped in a fake minimum or a minimum that's not very deep, and the minimum corresponding to the native state of the protein is very deep, and it's difficult to get trapped. Whereas sequences that are definitely randomly generated sequences, or that those that haven't been exposed to evolutionary pressures, can have these minima that are not as deep as the native one, but are pretty darn deep. And so the roughness of the landscape is much larger than the smooth one on the left. Now, to some degree, this idea is still a little bit vague. We don't have the full landscape for, that has been experimentally characterized for large proteins, but this has been done definitely computationally, but even on even portions of the landscape in some experiments.
Corey, I have a sort of random thought mostly. The energy landscape that you showed seems very similar to the loss landscape of a neural network, just a random comparison. And I was wondering, like there have been tools like AlphaFold that have used sort of neural networks to deconvolute and find structures of proteins. I wonder whether they use similar logic of energy landscapes, which are very in parallel to the loss landscape of a neural network.
Yeah, so that's a great question. I would say that this idea of smoothness and roughness is general. It's not general to proteins. It's been used as a explanation for why proteins, can find their native states. But it's generally called a glassy or a rough landscape. And in general, sort of all neural net problems fall into these classes, or fall into this rubric of explanation. I would say that I am going to talk a little bit about AlphaFold and sort of using neural nets, but I would say that, at least to my knowledge, there hasn't been a good physical explanation for why AlphaFold has solved the rough landscape problem.
Mm.
Or if it's just that the sequences that AlphaFold is folding are all smooth.
Interesting.
You see what I'm saying? Like AlphaFold has done some good work in finding in folding proteins,
Right.
but it could just be that for those particular proteins, they're not that rough.
Right. Got it. Thank you. That's very interesting. Thanks for that insight.
So given that AlphaFold has been brought up, I think it's a good well, it's fortuitous that we're now going to be talking about CASP. So CASP is the critical assessment of protein structure prediction, and it's a competition that has been running for at least 20 years, maybe 25 years. And what they do is they'll release a sequence or a set of sequences or targets that where the structure hasn't been released publicly. And so then they'll give the sequence to the community, and the community can use whatever techniques are available. So it could be neural networks, it could be lots of homology modeling coupled with molecular dynamics, any, a whole spectrum of machine learning approaches, whatever. And so you're given a little bit of time, and then you produce what you think the structure of the protein is. And so so then you submit your structure, and then since they have the gold standard, they have the actual structure, you can basically compare the computationally generated model with the right answer. And so one thing that's reported is what's called GDT. It stands for Global Distance Test. And it's essentially like a root mean square deviation of the positions. So you can basically, you have in the model a position of one of the atoms, or you have the positions of all the atoms, and then you have the positions of all the atoms in the real protein structure, and then you subtract them, take the square and sum over all the atoms, and then you can get a root mean square deviation. And then you can basically normalize that root mean square deviation into a score that goes between 0 and 100. It's a little more complicated than that because it involves alignment, and it involves how various weightings of different sets of atoms. But basically, it's a reweighted root mean square deviation of the atoms in let's just say it's the C alpha atom. So it's basically one atom per amino acid. And so 0 is a bad score. That means that basically all the atoms were in the computational model were incorrectly placed, and 100 is a perfect score. All the atom positions in your model exactly match all the atom positions in the target. And so the left plot is the GDT, the scores for the computational models for a bunch of targets. The targets sort of are indexed on the horizontal axis by the numbers of amino acids in the target. So some of the targets were small, around 100 amino acids, whereas other targets were large, greater than 600 amino acids. And then the vertical axis gives you the score for those models that were submitted. And those are models that are submitted by multiple groups. And each group can actually submit multiple guesses. So say on target number 1, which is 100 amino acids, say I'm a group, I can submit like 5 models, and then a bunch of other groups can participate and they each can submit 5 models. And here I'm just showing the scores of all of those models that are submitted. And so one thing that you see is that there's a big range. Some scores are 15, and then other scores are 80. And the thing that I'm not showing here is that you could say, well, maybe there's just some really bad groups out there, so let's just kick out the groups that are all in the 20s and don't show those. But what can happen as well is, remember, I was allowed to submit five models, and some of the models that I submit are 80, but also some of the models that I submit are 20. So it's not true that all the models that I submit for even a single target all get a high score. And so that's going to be what's called protein decoy detection. And I'm going to talk about more of that in the next slide. So sort of remember protein decoy detection. Now, there was a lot of hubbub or news related to AlphaFold. And so I can talk about it more, but basically what AlphaFold is, it was able to get really high scores, so between 80 and 90 on some of its targets, but it wasn't able to do that on all of the targets. So I would say that, and you sort of see that here, that definitely getting 90 is very rare. And so AlphaFold was able to do that more often, but there still are a lot of targets where AlphaFold did not do well. So even though AlphaFold is doing well, there's still a lot more sort of learning to do. So one thing that we've been focusing on in my group is the core of proteins. Remember I said that different amino acids, sort of behave differently. Some are non-polar, some are polar, some are charged. And so what we've been focusing on are amino acids that occur in the core. And what I'm showing you here is the correlation between the GDT of models and how well they place core residues, so atoms and core residues. And so what you see is it seems like there's a high correlation between high GDT and being able to place core residues in really well. So if the core residues have an RMSD of around 1 angstrom, then the GDT is already 80. So this suggests, and I'm going to sort of fill it in with subsequent talks of mine, is that it seems like that the core could act as a template, that if you place the core residues correctly, or if you could predict which amino acids are going to be in a core, then maybe you would get the rest of the protein somewhat for free because you're getting a GDT of 80. And there are some neural network approaches which actually do that. They focus a lot of their attention on being able to predict what amino acids are going to be in the core, and they can get pretty high scores.
Oh, can
Okay,
I?
so yeah, go ahead.
Yeah. So by core, do you mean the R group?
Say it again. By the what?
By the core, do you mean the R group?
Oh yeah, good question. So we're going to get into great detail, but sort of yes, but sort of no. So the R group is the side chain, and so it's basically does the side chain want to be next to a water molecule? And so based on the side chains, we group the amino acids into these 3 groupings: non-polar, polar, and charged. And you don't have to memorize all of them, but I think it would be good for you to sort of look at that slide for about 30 seconds and sort of remember, which amino acids are non-polar, which amino acids are polar. Sort of like leucine and isoleucine are non-polar, alanine's non-polar, serine and threonine are polar, and charged residues are like arginine. But what we're going to do, so then there's a high correlation between the non-polar residues, which are residues that don't want to be next to water molecules, and being in the core, because that sort of makes sense, because if you're in the central region of the protein, you're never going to see a water molecule because you're shielded from the water by the rest of the protein. And so we're going to talk about what specifically core residues means. It basically has to do with, do you ever interact with a water molecule? So if you interact very frequently with a water molecule, you're non-polar. I mean, you're polar, and you're a surface residue. Whereas if you never interact with a water molecule, you're a core residue. You're likely a non-polar residue. Did that help?
Yeah, that makes sense. Can I ask another question? So
you can ask as many as you want.
How does the host know that their structure is the correct one?
Yeah, so they don't. And in fact, that's called protein decoy detection. So whenever you're, whatever method you're using for predicting protein structure, like a neural net some type of classification or or molecular dynamics, there'll always be some type of scoring function. With the molecular dynamics, it's like the energy function I was talking about. So in that case, you typically want the lowest energy structure. Or in a neural network, you may have some cost function or loss function. And maybe if it's a cost function, you pick the lowest one. So you're always going to have some optimization algorithm to generate these structures. And you'll typically pick the lowest one, right? Because you think your method's right. But the question is, you never have nature's energy function or nature's cost function. And so the only way, the only gold standard way of determining whether your model is correct is to compare to the real structure, the experimentally determined real structure. But that gets me into the next slide, which is what's called protein decoy detection, which is a slightly simpler, I don't know if it's simpler, but it's a recast way of protein structure prediction.
Okay.
So now that we have GDT, so now we have 25 CASP competitions and we have thousands of models. We can now do a type of classification problem where we have the models and we have their GDTs, and we can say, well, which of the models turned out to be good GDTs. And so that's what's called a protein decoy detection problem, where you're given a model and you want to ask, is it a good model or is it a bad model? You see what I'm saying? It's slightly different. So in the first case, you're just generating a model and you want it to be good. But then since we've gone through these competitions and we have thousands and thousands of structures, you can say, well, I have lots of models and I have their GDT, and now I can do classification problems and try to predict the GDT given the model. And sort of these are the results from the current state of the art. And it's sort of good news, bad news. Like there is a correlation, but there's still a lot of false positives and false negatives. So the true GDTs on the horizontal axis, the predicted GDTs on the vertical axis. And so there can be a lot of models that were really bad structures, but the classifier thought they were really good. So that's in the upper left corner. And that seems like a much bigger problem than the opposite problem, where there are structures that have really good GDT, but the predicted GDT was around 40. So we're currently working on this and trying to understand why the classifiers were fooled. One big problem that's still out there is it's still really hard to predict which amino acids will be in the core. So, sorry. Okay, so along those lines, just to give you a little more insight or, so in general, what drives protein folding is a lot of different interactions. So there could be explicit charge interactions between residues, van der Waals interactions between residues, that's like dipole. And there could be this hydrophobicity effect, which involves the solvent. Like, do residues like hydrogen bonding with the solvent or not? There could be issues surrounding entropy. Do I get an entropy gain when I'm considering both the solvent and the protein? That's a really hard question to address. But based on a lot of solution-based studies, but also protein folding studies, there are ways to associate hydrophobicity indexes with amino acids. So one way, which is sort of a more numerical approach, is to just look at folded proteins and then calculate the probability that a particular amino acid occurred in the core. So I have a bunch of alanines that occur in proteins, and then you can just go look into experimentally determined proteins, and you can ask, do they occur on the surface? Do they occur in the core? And then you can assess a hydrophobicity index with that fraction. So related to that, just look at panel A, the top panel, and just focus on, say, the gray histogram. I think that's on the leftmost rectangular histogram. And so that's those gray boxes represent experimentally determined structures for globular proteins. It's abbreviated DUN 1.0. The 1.0 refers to the resolution. So they're very high resolution X-ray crystal structures of globular proteins. And what we look at here is what's called R-SASA, which is going to be defined on the next slide. It's called the relative, which is the R, SASA, which is solvent accessible surface area. So solvent accessible and then surface area. So surface area is given in typically an angstrom squared because it's an area, and you basically look for the area of an amino acid that can be accessed by a water molecule. And so I'll tell you a little bit more about how that's calculated, but solvent accessible surface area is typically angstrom squared, and then the relative solvent accessible surface area is a dimensionless number, so it doesn't have any units. And so you normalize it by the surface area of the entire residue. So I'll tell you more about that, but in the next slide. But what we do here is we use a cutoff for the relative solvent accessible surface area that's very small. So it says that water molecules basically can never get very close to these particular amino acids. And so what you see are these typically hydrophobic or nonpolar residues showing up. So alanine, valine, glycine, leucine, isoleucine are very hydrophobic residues, and you're seeing that they don't have very much solvent accessible surface area. So they're typically occurring in the core's central regions of proteins, whereas charged residues like arginine, lysine, glutamine, and they have lots of solvent accessible surface area, and so they're never occurring in the course, or very infrequently occurring in the course. So here's a schematic. It's not very good. It's in 2 dimensions. The weird shaped particles are representing amino acids in 2 dimensions. So I have different types of amino acids, and this glob here is supposed to represent the folded conformation of a protein. And what I'm doing here is trying to calculate the solvent accessible surface area. So the white part of the screen on the outside of this protein is representing regions of space where water molecules can sit. And so you see the cyan shaded region in the upper left there is the exterior surface of this amino acid. And you can imagine that a water molecule can basically sit there and interact with this exterior region of this amino acid. And so if you calculate it, in this case, it's like a perimeter because it's in 2 dimensions, but in 3 dimensions, it would be a surface. And so that cyan region represents the solvent accessible surface area of this particular residue. And then the rest of the perimeter of that residue would be the solvent inaccessible surface area of that residue. And so then, as I mentioned on the previous slide, because residues have different sizes, the absolute value of the solvent accessible surface area sometimes can be confusing because amino acids have different sizes. So if a residue tends to be very large, even if it had a relatively small solvent accessible surface area, you could be tricked because it's so large. So we think a better measure is to calculate the relative value. So that's why in the lower portion of this slide, we say the RSASA, we have the relative solvent accessible surface area is the solvent accessible surface area of a particular residue divided by the solvent accessible surface area in the absence of the protein, where the whole thing can be accessed by a water molecule. So you divide by the entire surface area of the residue. Just a quick note is that here I'm just saying it's the complete exterior surface. That can be a little bit complicated because water molecules can intercalate and go into the inside. So what we do is we don't just do the exterior, we look wherever a water molecule can be placed inside the protein without overlapping an atom of the protein. So we don't just get the sort of easy outside of the protein, we actually go in and see whether water molecules can go in sort of the interior layers of the protein to determine the solvent accessible surface area. And so we're going to be talking about the relative solvent accessible surface area throughout these set of lectures. And so the solvent accessible surface area, the relative solvent accessible surface area, RSASA, is a number between 0 and 1. And so a value of 10 to the minus 3 or 10 to the minus 2, so 0.001 or 0.01 is a value that's taken on by core residues, and anything larger than that is a non-core residue. And as you approach, 0.5, you get to be what's called a surface residue. So just, I'm not going to go through all of this right now because we're going to get into very, very detailed discussions of secondary structure. But I just wanted to get it on your mind now, is that there is secondary structure in proteins. So as I mentioned that proteins will take on very specific structures, and we know a lot about that structure by experimental characterization. So this is typically by X-ray crystallography, NMR spectroscopy, both liquid state and solid state, and then more recently electron microscopy, or cryo electron microscopy. And so that's how we know in detail about the structure of, in the cases of high resolution structures, we know basically the positions of the atoms in many cases to 1 angstrom resolution or better. And when we know that, we basically know the structure of the protein. And so the secondary structure talks about how groups of amino acids, say 2, 3, 4, 5, 6 amino acids, sort of collectively situate themselves to form structures. And so the very common secondary structures are various types of helical conformations. So for example, the most common helical conformation is an alpha helical conformation. And then there can be other types of conformations of amino acids where you can have beta strands, which will be, groups of 3, 4, 5 amino acids take on a particular conformation. And then those beta strands can self-organize into a sheet-like structure and form beta sheets. Same thing with alpha helices. Alpha helices can sort of collectively arrange themselves to form, say, bundles of alpha helices. And so you can talk about tertiary structure, where you can get these sort of groupings and bundles of particular types of secondary structure. But one idea is that you can look at secondary structure sort of on a per amino acid, or at least a few amino acid level basis. And so, for alpha helices, they tend to form, they have these backbone. So I'm going to get into it in much more detail, but what we're going to talk about is what are called backbone dihedral angles and side chain dihedral angles. And backbone dihedral angles talk to you about, well, what are the relative angles and positions that the nitrogen, the alpha carbon, the carboxyl carbon, and the carboxyl oxygen are making with each other. And if those, 4 atoms are making specific types of relative conformations, then they can form an alpha helix, a beta sheet, or other secondary structures. And I'm going to talk in much more detail about it, but for alpha helices, the backbone dihedral angles are phi and psi, and they typically have this -60 degrees, -45 degree conformation. And for beta strands, they have this -135 degrees and 135 degree conformation. And then if multiple amino acids are taking on those particular backbone dihedral angles, then you can form these secondary structure motifs. Now, with the alpha helices, you need a few to form a helix. So in an alpha helix, each amino acid makes a 100-degree rotation in an alpha helix. And so you need 3.6 amino acids to make a complete turn. And so you need between 3 and 4 amino acids to sort of get back where you are in the orientation of the alpha helix. When you form an alpha helix, the side chains stick out of the alpha helix, so they're not sort of condensed on the interior. And there tends to be hydrogen bonding between the amine group and carboxyl group. Now, there's been a lot of debate about a chicken and the egg, whether the hydrogen bonding that occurs in the alpha helix structure is necessary for it to form, or whether it sort of stabilizes after. It's still sort of an ongoing question. Maybe it's a little bit of both, but they're just for your edification. If you stick a bunch of atoms in a cylinder to pack them very efficiently, they tend to form helical structures. And so that's sort of just their geometrical influences for sort of close packing of spherical atoms that induce a helix formation. So there's both sort of close packing geometry that's playing a role, but then also you could get an extra benefit of bonding because just the way the side chains sort of interact with the next carboxyl group in the alpha helix structure. The same thing can be said with beta sheets and beta strands, that to what extent is geometry playing a role? To what extent is hydrogen bonding between the beta strands causing the structure to form? Another thing to note is the propensities that I list at the bottom, that you can just sort of do a data mining exercise, and you can go in and look for alpha helices and experimentally determine structures. And people have found that methionine, alanine, leucine tend to occur in alpha helices and experimentally determined proteins. And the same thing for beta strands, valine, threonine, tyrosine, and tryptophan tend to be a beta strand or beta sheet common amino acids. Just to give you an idea of the order of magnitude. So here I show, I think it's as of like 2 years ago, but it's the number of X-ray crystal structures histogrammed as a function of resolution. So this is, if you look in the protein data bank and search for X-ray crystal structures, so you'll find, so there are 2 bits of information you can get from this. So first, the total number of structures is approaching 1 million, because you can see we're getting up to 10 to the 5. Sorry, it's probably a few 100,000, because we're getting sort of almost 10 to the 5 in each of these bins. So a few 100,000 X-ray crystal structures, and you see that we're getting lots of structures that are 2 angstroms and below. And so there's lots of nice data to analyze. One thing that's not extremely well known, and I wish it were, and I'm hoping, is that we don't really know how well we should know X-ray crystal structures. That is, how well do we need to know a protein structure to really identify whether it's the right structure that can perform a specific function, or whether it is the right structure that can bind to another protein. And so we sort of have intuition that it's not 5 angstroms, but we don't have good evidence that we need to know everything to 1 angstrom and below to know whether it's going to bind or perform a particular function well. The other thing we don't know, which I'm trying to emphasize more in these slides, is that all of these X-ray crystal structures that I'm showing are in non-physiological environments, very low concentration, sometimes, in almost all cases, not cellular environments. So we don't know how different the cellular structures of these proteins differ from these in vitro environments. So we know the in vitro structure to a certain resolution, but we don't know the in vivo structure to this resolution. So keep that in mind. So given all these high resolution protein crystal structures that are in vitro structures, we can sort of carefully analyze their structure. So one thing we can do is look at the distance between every bond and the bond angle between every 3 set of atoms. So between every 3 set of atoms, there's something called a bond angle between covalently bonded atoms. And between every 2 bonded atoms, there's a bond length that we can discuss. So what you can do is go in and all those high resolution X-ray crystal structures and just measure all of the backbone bond angles and all the side chain bond angles. And so For example, we saw in the first slides on amino acid structure, that there's a nitrogen that's bonded to a C alpha, and then there's a C alpha that's bonded to a carboxyl carbon. Then the next one is that there's a carbon that's bonded to, there's a, so this one, so this one's kind of interesting. It's a carboxyl carbon that's bonded to an alpha carbon, which is then bonded to a side chain carbon. And so the nomenclature for side chain atoms is you start at the alpha carbon, and then you add additional Greek symbols as you move away from the alpha carbon. So the next in a side chain, if a side chain has an additional carbon, it's the beta carbon. And so then the next one would be a gamma carbon. So I'll show you that in a second. So this is just doing this again, this is a carboxyl carbon that's bonded to an alpha carbon on the backbone that's bonded to a side chain beta carbon. So that particular bond angle involves both backbone and side chain carbons. And then this next one is a fully backbone bond angle. So it's an alpha carbon that's bonded to a carboxyl carbon, which is then bonded to the oxygen of the carboxyl group. And then this next one is interesting. Oh, everything's interesting. It's a C alpha carbon that's bonded to a carbon on the side chain, which is then bonded to the next carbon on the side chain, the alpha carbon. And then this can go on. You can, after the gamma carbon, you can have a delta carbon and a delta 2 carbon for leucine and isoleucine. So there are two things I wanted you to get out of that slide. It's the nomenclature for the sort of heavy atoms, and then also that the bond angle distributions are fairly narrow. So for example, a lot of them are focused around 110 degrees, between 110 and 115 degrees, where you would, which is what you would expect for these covalently bonded carbons. And the root mean square deviations for these distributions are typically around 5 degrees. And so that's pretty well defined. But there are a few that are slightly bigger that go between 5 and 10 degrees. There's a very common one. It's this nitrogen C alpha carboxyl carbon, which typically can vary between 5 and 10 degrees. That's typically called what's called a tau angle. And sometimes you have to track that because that can affect protein structure. But what I wanted to get at was that to some degree, these bond angles are fairly fixed. Since they don't vary by much, we can consider them to be basically determined by what we call protein stereochemistry, that all proteins sort of share this feature, and we don't treat it as a variable. We treat it as something that's given by the nature of covalent bonds. Similarly, we can, these are the bond lengths that occur between every two covalently bonded atoms. And here I show you a few examples, like between an alpha carbon and a carboxyl carbon, between the carboxyl carbon and the carboxyl oxygen, so on and so forth. And these are even much better defined. They typically vary between 0.1 or 0.2 angstroms. And for all intents and purposes, for most models, are fixed at the average value. And so again, we say that these degrees of freedom are fixed by the nature of covalent bonds and protein stereochemistry. The things that aren't fixed are what are called dihedral angles. And dihedral angles is a variable that is defined by 4 atom positions. So I just gave you 2 examples of variables that are defined by either the positions of 2 atoms or the positions of 3 atoms. And now we're going one more up the chain, a variable that's defined by the positions of 4 atoms. And the way that you think about it is, here I have 4 atoms with kind of funny names. One's called I plus 1, AI minus 1, and AI minus 2. It's just a way of denoting the positions of 4 atoms that are bonded together. And what you can say is that any 3 of them, and so there are 2 groups of 3. So if you have 4 things, there are 2 unique sets of triplets of those. So if you have 4 things, A, B, C, and D, or AI plus 1, AI minus 1, and AI minus 2, there are 2 distinct groupings of those. One is AI plus 1, AI, and AI minus 1. That's one unique triplet. And then the next unique triplet is AI minus 1, and AI minus 2. And the reason why I focus on the triplets or 3 things grouped together is because those form a plane. If you have the positions of 3 things, you can uniquely define a plane in 3 dimensions. And so those 2 unique triplets define 2 planes. You know what's nice about a plane is that a plane has a unique direction, which is the vector that's perpendicular to the plane. So basically, a plane can be characterized by the perpendicular or the normal direction to that plane. It's basically how the face of that plane is pointing. And so since 2 planes have 2 unique directions, you can take the dot product of those 2 vectors, and that defines an angle. And that angle is the relative orientation of those 2 planes. In this sort of silly notation, I say that the vector that describes the orientation of plane 1 is called pi 1. The vector that characterizes the orientation of plane 2 is pi 2. I take the dot product of those 2 vectors, and that gives me the cosine of the angle. And that angle is called a dihedral angle. It's completely analogous to how you define a bond angle in the previous slides. Is that how you define a bond angle is basically between 2 atoms. And so you take the dot product of the vector that defines, the 2 unique groupings of 2 atoms. You take their dot product, and that defines the cosine of the bond angle. Okay, so these dihedral angles can either involve backbone atoms or side chain atoms. And if they exclusively include only side chain atoms, it's called a side chain dihedral angle. If it involves backbone atoms, then it's called a backbone dihedral angle. The backbone dihedral angles we've already talked about. I've introduced today. They're called phi and psi. And so we'll talk in much greater detail on Wednesday. And then I'll talk further about the side chain dihedral angles. I'll just quickly mention that there's a way to find out how many dihedral angles you have if you have a polymer that has a certain number of bonded, of a certain number of atoms that are bonded together. So if you have n atoms that are bonded together, then you have n minus 3 dihedral angles. So this obviously only works for greater than 3 bonded atoms, because I just said that you needed 4 atoms to define a dihedral angle. So if you have 4 atoms that are bonded together to form a polymer, then you have one dihedral angle. If you have 5, you have 2 dihedral angles, and so on and so forth. So this is a way to figure out how many dihedral angles you have. So with that, I will finish today. So it's 2:15. And so on Wednesday, I'll talk specifically about how to define the backbone and sidechain dihedral angles, because that's how you actually discuss protein structure. Are there any questions to round out today? I'll wait basically about 10 or 15 seconds, and if I don't hear any questions, then we can just join together again on Wednesday.
Corey, could you go back like a few slides? Yeah, maybe the next one. Next. Sorry, the forward. Yeah, this one. It seems like the distribution is not normally distributed. Is that illusion or?
I think it may be an illusion. It or not. Yeah, I have to remind myself whether they are. I'm trying to think if there's a good reason both ways. I mean, in general, because so on the one hand, I would expect, since they're somewhat random variables, and I'm looking at large numbers of these random variables, I would expect them to be Gaussian. But I don't remember here how many I picked to create this distribution. And so let me just see if, I don't want to take too much of your time, but. Can you still see my screen?
Yeah.
You don't have to look at this. I just wanted to quickly see if I had another distribution.
Sure.
Okay, I'm not going to be able to find it. My first guess would be that they should be Gaussian, but let me dig some better ones up, or at least ones that I know how many samples were included, and then I'll bring it next time.
That sounds good.
Any other questions? Okay, it's been a pleasure. I'll see you all on Wednesday.